Hi, today we're going to talk about molecular genetics. So the entire basis of molecular genetics is to look at the DNA, and also we'll be looking at what comprises each individual DNA. The second segment of the video will be looking at what is known as the central dogma of DNA, and lastly is how genetic engineering is being utilized in the regular fields these days in terms of medicine or even agricultural. So right here, the simplest units of DNA is made up of nucleotides over here. Okay, so every single nucleotide is made up of 3 different parts, and these nucleotides are repeated throughout the millions of base pairs in the DNA structure. So each of these 3 parts, we have got a phosphate group over here. Okay, a 5-carbon ring sugar, meaning to say that this sugar itself has 5 carbon components to it, which we will look at. It also has a nitrogenous base, and there are 4 types of nitrogenous bases. Okay, so let's look at the sugar first. So there are 2 types of sugars. One is DNA, which is called deoxyribose, and the other one is called ribose, just RNA itself. You can see that over here, the structure of the 5-carbon ring, okay, starting from the right hand side, the top part here, you have got the first carbon, second carbon, third one, fourth carbon, and the 5th one, which is on top side of it. Okay. So when we look at the second carbon itself, the main difference between the DNA itself and the RNA, we can see that there is a hydrogen on the second carbon, but on the ribose, the RNA itself, there's a hydroxyl group over here. So the hydroxyl group gives it the oxygen, but in the DNA there is no oxygen, which is why it is called deoxy, meaning to say one of the oxygen is removed. So this 5-sided sugar is what we call a pentose sugar. So pentose simply because it is 5-sided here. So bases, nitrogenous bases that we spoke about earlier on, there are 4 types. So 4 of these bases, when we look at just the DNA first, we have got ATGC. Okay, so we have got A here, T, G, guanine, and cytosine. Okay, now do take note that over here, in terms of the spelling for adenine, a lot of students make this mistake when they did the spelling for adenine. Do not spell it as adenosine. So this one is the spelling that is in the energy currency when we did mitochondrial aerobic respiration. So this one is definitely not adenine, which is a common mistake that students tend to make. So just take note of this, it's not this. They are totally 2 different molecules of their own in 2 respective components of the cells. Okay, so once we understand that there are the 4 types of nitrogenous bases, A, C, G, T over here, A and G is what we classify them under the group of purines. Okay, whereas for C, cytosine, and T for DNA itself is classified under pyrimidines. So we can see the difference over here. Purines have 2-sided double rings, 2 rings here, but pyrimidines, we are only looking at 1 ring. Okay, now what about uracil? Uracil, later on when we look at the mRNA component or the RNA itself, there is no thymine in RNA. So instead of thymine in RNA, the thymine is being replaced by a uracil in this case. So when we talk about DNA, we know that it is double-stranded. Okay, so the structure of the DNA goes this way. So it is always 2-sided over here, which means to say that if I have got a set of nitrogenous base here, it needs to pair with a corresponding component here. So which parts are the ones that are being paired? Now A over here, adenine, as we saw earlier on, it is a purine. So a purine will pair with a thymine, which is a pyrimidine. Okay, so likewise over here, the C, which is a pyrimidine, will pair with a purine, which is a guanine over here. Okay, so same thing, G will pair with C or C will pair with G. Therefore, they are in a 1:1 ratio. So adenine and thymine, they are also in a 1:1 ratio. So if I know the concentration or the percentage of adenine, take for example, the percentage of adenine is 20% in the DNA. So which means thymine also will make up 20%. So the remaining 60% will be split evenly between the guanine and the cytosine, making it a 30% and a 30% over at cytosine. Okay, so this one is for the DNA component. So over here, you can see that these are dotted lines components here. So I have got thymine looking at DNA and adenine looking at the DNA component itself. So thymine and adenine, they pair with a double bond over here, whereas cytosine and guanine, they pair with a triple bond. So if we were to write it out, it will be C with a triple bond of hydrogen bonds to guanine in this sense. Okay, so this one is the structure of the DNA. We can see that it is double helix. So this part here is also double helix because it is bonded together. So earlier on, we looked at how the structure of the DNA composed of these three components, whereby we have got the phosphate group, we have got a sugar, and we have got any one of the four nitrogenous base. Okay, so this is where we look at it. Over here in yellow, we have got the phosphate groups. Okay, likewise, on the right hand side, we have got the phosphate groups, and all of these, they are joined by the 5-carbon pentose sugar component, and these ones, they are covalently bonded, meaning to say that the bonds are very strong. Okay, and these ones, they make up what we call the backbone of the DNA molecule over here. Okay, so now if we were to look at the structure of here, remember just now we look at the pentose sugar in terms of the naming system, the numbering system. So here I have got the first carbon, and the 5th carbon is right at the top over here. So likewise, when we look back over here, if I were to zoom it in, okay, you can see that I have got the first carbon, 2nd carbon, 3rd carbon here, 4th carbon, and this is the 5th carbon. So in this area, the phosphate group is being binded covalently with the 5th carbon, and therefore we call it the 5' end. Okay, so 5' end meaning to say that this part is bonded covalently with the 5th carbon of the pentose sugar. Alright, so 3' over here is because here I have got the first carbon, 2nd carbon, and it ended off with the 3rd carbon, and therefore it is the 3' end. Okay, so let's look at here. Now this part, we are starting off with the 3rd carbon component. Okay, so this one is the 5th carbon, this one is starting off with the 3rd carbon, so this is what we call the 3rd carbon, and this one, same thing, this is the 5th carbon, so it's the 5' end. So what we can see from here is that the 5' end here starting, and this is the 3' end starting, which is why DNA is what we call anti-parallel. Okay, so just let me write that down. So DNA is in an anti-parallel structure, okay, because of the difference in the 5' to 3' configuration. So these middle parts here, and when you do DNA replication, you actually realize that these ones, the hydrogen bonds by themselves, they are weak enough to open up the strands. But when you have got millions of base pairs being joined together by hydrogen bonds, they are strong enough to withstand external attacks, okay, and to prevent it from opening easily. Okay, so when we looked at condensation and how it forms together, so this part I have got my pentose sugar, and over here I have got my phosphate group of another nucleotide. So this whole component, we have got one set of nucleotide together with the base. Same thing, one entire set of the nucleotide with another base. Okay, so in this section whereby you have got two bases coming together, it will form what we call a condensation. So condensation is the loss of water during the formation of a covalent bond. You will see that under the topic for digestion as well. So water is being lost, H2O. So once it is lost, this particular phosphorus here, the element itself, will bind covalently with the oxygen on the hydroxyl end of the nucleotide base on the other side. So that will get you a covalently bonded new structural backbone. Now, when does this take place? This normally takes place when under the topic for replication, DNA replication itself. Okay, so this part, when we look at bonding itself together, eventually at the end of the day, you have got a long series of nucleotides, and each of these bases, nitrogenous bases, they are paired in complementary structure. So T is always bonding with A. So likewise, A will be with T over here. So if I've got a G, I will definitely have a C. Likewise, if I've got a C, I will definitely have a G as well in a triple hydrogen bond. So because of that, this variation in the sequence gives rise to genetic variation, which is why no two people are identical unless you are identical twins. Okay, so the next part here, we will be looking at the double helix itself and how it contributes to the genes.